Okay, so in this week's lab, we are doing a writing workshop for the results and discussion sections specifically. And we have, before we went to breakout rooms, as a reminder, we broke the outline into 3 major chunks. And these are essentially sets of data. So the first set of data is the Bradford assay standard curve, which produces the equation, which allows us to calculate the concentration of each of our protein lysates that we're going to be analyzing. The second set of data are the staining patterns from a Ponceau stain of the nitrocellulose membrane. And then the third main set of data is the actual gold at the end of the rainbow, the treasure of the entire lab report. So what this really does provide us with the answer to our original research objective. And so we are going to be talking about these sets of data. First, we'll talk about the results, and then we'll pair it with the complementary discussion that should kind of pair up with that, right? And so that's how we're going to start this whole thing. So let's go ahead and do that. Yeah. Alrighty. Okay, so everyone should have their notes out, and I'm just going to pretty much ask questions, and you're going to be actively participating in the chat. This is being recorded, so people's participation, the transcript of the chat does get recorded, so I get to see who in fact is not participating. So there's 24 people here. I should see 24 responses when I ask for everyone to participate or to enter in an answer. And just remember, I don't care if the answer is right or wrong. This is the time for us to make the answers right, make the answers more specific, iron out all the maybe mini misconceptions. So at a minimum, there's 6 rooms. So when I do ask for person number 2, person number 2 from each of the rooms is going to contribute to the chat. So that means at a minimum, at any one given point, I may only see 6 responses, but please stay engaged. Please enter in when I ask you to. Okay, so we're going to start really simply, and we're going to say, do not press enter, but person number 1 in each group, please enter in the topic sentence for this first topic. Do not press enter. Okay, now press enter. Okay. Okay, so I like how this is starting. To accomplish a certain goal, these things were done. Okay, I wouldn't start putting the results as a topic sentence. That's an important detail, but it doesn't go in the topic sentence. Okay, so this thing was done so that we can accomplish this. Okay, so that we can compare the amounts, the concentration of each protein was determined. Okay, great, we're talking about protein concentration. Good. So I'm not going to rewrite any of these, but I'm just going to put down on this class discussion document the things that I think should go in a topic sentence, which would set up the rest of the paragraph so that all of the supporting sentences can easily support this topic. So, so you have the goal, which is determine the protein concentration, the protein lysate concentration. So if anyone has it vague and just says to determine protein concentration and don't tell me of what and don't tell me anything besides it just being protein, I would add a little bit more detail. So in terms of detail, you can say, you can make it a little bit. So how would you, oh, somebody actually has this. Pisativa, great. Thank you. So to determine the protein concentrations of pea sativum, and then grown in different light conditions, a Bradford assay was performed. Boom, it's done. And that's a really good topic sentence, because what that sets you up to do is the next following sentences in this paragraph, you can establish how the plants were grown, right? This is the time you want to do it because you're setting up the whole results section. So we don't need to go into this. I know you know it. So actually, we're not going to take a long time to do it, but this is a question for everyone. What are the three conditions in which the plants are grown in? So go ahead, just phrases are okay for now in the chat. Okay, I'm gonna write them as I see them. Okay, great. Okay, so you start with this topic sentence like, hey, reader, we're going to be figuring out concentrations of this plant. The plant was grown in different conditions. We're doing this by doing a Bradford. Oh, you want to know what conditions the plants were grown in? Okay, cool. Here's the next sentence. These plants were either grown in a 12-hour photo period, and then this is when in parentheses you can write the phrase positive control, okay, comma, in absolute darkness or total darkness or complete absence of light. In parentheses, you write the phrase negative control. You're not using symbols, you're not using the plus sign, you're not using abbreviations. This is the written results, you're writing things out. You put the phrase positive control in parentheses, negative control in parentheses. Why? Because that is the less specific phrase that you can use to describe these samples. The most specific phrase is the most important, which means it does not go in parentheses. Okay, and establishing a positive control and establishing a negative control, I really don't think it's necessary to put experimental sample in parentheses. If it's not a control, it's pretty obvious that it's something that you're testing. Okay, so now you've discussed the three growing conditions. If you just put 60 minutes of light and don't describe when those 60 minutes occurred, that's an issue because these plants were grown over the course of, I think it was either 14 to 17 days, I can't remember, but there's a lot of 60-minute increments in that time. So make sure you delineate when those 60 minutes were. Okay, good job. Okay, so the topic sentence says it also is going to have to do something with the Bradford assay. So let's go with person number 2. Just go ahead, don't push enter yet. I don't want full sentences. I don't want blocks of paragraphs. I want phrases. So when you are writing the results of the Bradford assay, what are some of the specific phrases that you have included? And remember to keep them in chronological order. So how did you obtain these phrases? Does it make sense with how the experiment actually happened through time? So again, this is person number 2. Phrases that you've included that are about the Bradford assay results. Don't push enter till I tell you to. Separate your phrases with a comma. Enter. Okay, so okay, so this starts out with the end result, which is at least one of these comments does. You can't start with the equation because there's definitely some important phrases that allowed you to get to that equation. So if we're keeping in line with what is chronological order, so I'm going to come back to Hannah. You have some important things, but I think that needs to go a little bit later. Let's see. All right, so Ellery, great job. It doesn't require a huge sentence describing what it is, but just basically stating that a standard curve was prepared with BSA, I think is probably pretty important. BSA, do you have anything about, so one of the things I asked you all to do was look back at the feedback from the serial dilution molecular bio bootcamp, and I'm almost positive that if you did not write the dilution factor in your written results, I told you in the feedback, what is the dilution factor? Because that's important, because many dilution factors can be done. So yes, I think stating a standard curve with BSA at a DF equals 2 or dilution factor of 2 was created or made. Okay, so definitely you want your DF there. Okay. Okay, so please use previous feedback, because I didn't just feed it back to you. I didn't just do negative or positive feedback for back then. It's so that you can use it in the future, not just for the bootcamp assignments, but from that point forward, I expect you to remember and use that for the rest of the semester. Same way when we talk about polar bonds and covalent bonds, I expect you to use that the rest of the semester. Okay, so BSA curve was prepared at a dilution factor of 2. And so when you did this, let's see if anyone has this. Standard curve was calculated. Okay, so Bradford assay was used to generate the curve, the equation of the line. All right. Okay, if we're just thinking about the BSA standard curve, what are you basing this off of? So the standard curve is what? What is the standard curve? It's BSA versus what? Or can you come up with any kind of, besides the equation, is there any summary of data that you can come up with for this graph? Yes, it's BSA versus OD. Great. Anytime you plot something against something, are you not looking at a relationship between them? Thank you, Michael. Okay, a positive or a direct relationship was observed. That's important because the BSA curve pretty much is your control for this whole thing. So you better know what to expect. And so you expect a positive train. I mean, trend. I guess I'm predicting the train's going to come by anytime soon. Okay. So you mentioned what kind of trend it is. Okay. So the Beer's law, great, Madison. I think that could be mentioned in the discussion, because that is your prediction and it's based off of Beer's law, which indicates the relationship between concentration and absorbance is direct, right? So direct because Beer's, or based on Beer's law. Okay, so results, you're stating the trend, and in the discussion, you're going to explain what you predicted, if you saw it, and what that prediction was based off of. Great. Okay. So now just like imagine you're doing this in the lab, right? So you created the BSA curve, you did it at a dilution factor 2, you stuck all those samples in the spectrophotometer, you got the data, you made this really nice graph. This is one sentence. Okay, after you did the graph, I think you can still even concisely write this in a sentence. You calculated the equation, and you people are telling me that the equation is 3.65x, right? So these are your main data points, this and this. Very simple, very clean, very concise. You're not explaining why, you're not explaining the results, nothing like that. Very straightforward. Great. All right, so once you get that equation, then you say to me that you're going to use that equation to determine the concentration of each lysate. Making sure that it doesn't sound too redundant, too copy and pasty from the actual topic sentence. Okay, so now that we have this equation, and I think some people already did it, yeah, so now people said that some of the other phrases they use in this section, it's not really a formal section, but we're gonna call it a section today, is you actually list out the concentrations. And since you have already described the samples specifically, and in parentheses, you put positive control, I think now it would be okay to write down here the concentrations for the positive control, or there's a couple ways to say it. You can say something like, the concentrations were number for the positive control. You're writing all these things out. I'm just taking notes here. For the positive control, you're not using these abbreviations. Negative control, 60-minute light plants, however you want to say it, respectively. Okay, that's one way to say it. Or you can say, we calculated the concentration of the positive control to be this, of the negative control to be this, and of the plants exposed to 60 minutes of light to be this, period. That's it. So those are two ways that you can write the same thing. So this is option A. There's multiple options, but here's just two ways. We calculated the concentration of the positive control to be blah. Etc. Okay, whatever feels good for you guys. And there's, like I said, different variations. Now, here's my question for anyone, because I know that you were in a breakout room with some new people, and maybe some of you looked closely at the data, maybe some of you just are generally more naturally curious, but at this point, are there any questions or things that they saw about the data for the protein concentration or the Bradford assay that they were like, that seems weird? Anyone want to mention that? Or anything that they saw like, yeah, I see that. Okay, so Madison Yoakum, you said something was not as expected. Ashley saying, why is the concentration of the positive control much lower than the other samples? Negative control. Okay, so you saw something that was a little weird. Let's put it in a different color. Possibly weird. You're not going to say weird in the report. Okay, so there is a discrepancy, or not a discrepancy, but there is an obvious difference in the amount of, in the concentration that you obtained, right? And so what that means, because remember how this happened. So it's weird that the concentrations are so different. And you'll basically just be pointing this out, right? Maybe not on, if you want to use unexpectedly, that's fine, or interestingly, because to me this is kind of interesting. Interestingly, the concentrations for blah was much higher than the concentration of this. That's all you're going to say. It's just basically hint that it's weird. And then in the discussion, you're going to explain why you think that weird thing happened. Okay. So some weird things going on with concentrations. And so we're not going to go into why that is, because that's for you to use critical thinking. But I think it's important that we point this out, and I will give you some kind of starting points. And if you didn't think that was weird, then I encourage you to start using a more skeptical eye when looking at data. So remember, each sample, I gathered 1 gram of tissue. So if you're thinking that the concentrations being so different is something that is odd, then you are assuming that for every gram of tissue, no matter where you get it from the plant, that place in the plant is making the same amount of protein. So there is a prediction here. If you thought it was weird, then you're predicting that no matter where you get the 1 gram from, you are predicting that same amount of protein. Thus, you would have the same concentration of total protein. So this needs to be backed up with some kind of reference. Okay. Okay. And that's why you would think that that was weird. So if you got results, keeping in line with this kind of template, you state the prediction, you recap the results. So if we're talking about recapping the results, we'll say that the data indicated that, which one has it? Negative control. You recap the data. So that means the negative control has a much higher protein, I'm concerned, much higher protein concentration than the positive control. So there you are, recapping the data. And so even just by using the word like interestingly, that tells the reader that that's not what you expected, that the data does not support your prediction. Okay, so what does this mean? This means that this prediction is not true. So what it does mean is that it matters where you get protein from. That 1 gram of tissue mass, those cells in that tissue mass, they do not make the same amount of proteins. And then you might look up some research to kind of back that statement up, right? Back up that conclusion. You can say, oh, the data suggests that a gram of tissue from the stalk is going to, a gram of tissue from the stalk is going to produce more protein, or there's more protein in that gram of tissue versus a gram of tissue from the leaves. Okay. Yeah. So, and then you can say this is similar to what was seen for cactus cactobillus, where if you harvest tissue from the roots versus the spines, there is a different amount of protein. Okay, so, oops, let's see. Data did not support, so big fat no, and then you explain what that means. So it means that location matters. Location of tissue matters. And it also means that different parts of the protein, cells in different parts of the protein make different amounts of, cells in different parts of the plant make different amounts of protein. Okay? Different cells make different amounts. Again, this is not the whole focus of the study, so don't spend too much time there, but I think it's as simple as just saying that. Okay, so 1 gram, but remember that for some samples we got it from the leaves, from other it was leaves and stalk, branch, I don't know. Use the right terminology. Stem, okay. Okay, I'm not a botanist. Okay. Okay, so good job seeing that and having that be like, why did that happen, right? Cool, but that again tells us why it's so important to make sure that we start with the same mass of protein, not just the same mass of tissue and then get the concentration, but the same mass of protein. Because as we can see, collecting 1 gram of the stem versus 1 gram of the leaf is going to result in a starting material with way more protein versus way less protein. Okay, great. And that's it. That should not be like this very huge paragraph at all. So let's just, I won't do the sentence counting for each thing, but let's just do it for this one just to kind of see. Obviously you have a topic sentence. This explains how the plants were grown. This, I think here, can all be combined into one. Actually, I think this can be combined into one sentence. And then you have your results. So it's either one of these. If there's anything weird, there it is. You're not going to explain why, not in the results. You're just pointing it out like, hey, by the way, planting a seed. You'll read about it later in the discussion. Last but not least, you need a transition sentence. Okay, so if we look at what we're going from, we are going from plant concentration to ponzo stain. So the last thing we talked about was the concentrations of each of our proteins, and now we're going to red stain. So you can't just Back to the Future skip through time travel. You need to at least state what happened between here. How did we get from here to the red stain? So I think we've hit, I'm gonna turn my heater off. We have hit person 1, person 2. So this is going to be person 3 in the breakout room. Please don't press enter, but do type in your transition sentence or some idea of how you might flow smoothly from concentrations of protein to ponzo stains. Think about what the scientists did to get from tubes to a red stain. Don't push enter, so just go ahead and type it in. Okay, go ahead and press enter. Okay. And everyone should be reading these too. See if it's different, similar, what's the same, what do you like, what do you not like. Okay. So it looks like we're kind of going through time, like what happened next. We're not making a huge long, super long sentence, but we're just saying that once protein concentrations were determined, lysates were run on an SDS page to separate by molecular weight, or separate, yeah, the lysates were separated by molecular weight on an SDS page and then transferred to a nitrocellulose membrane for ponzo red staining. Great, that leads, oh my gosh, that's great, right? So, okay, so once you have the concentrations determined, you're going to separate them by molecular weight using an SDS page. Okay. And then you're going to transfer them to a nitrocellulose membrane for what? For ponzo staining. One sentence. And it flows so naturally into the next part. Oh, you're transferring to a nitrocellulose for ponzo staining? Cool. And guess what? Now I'm going to tell you all about ponzo staining. Okay, so this is kind of the template that you use every single time. I don't think in this situation things like 30 micrograms of protein are needed. That is definitely going to be written in the material method section. Okay, let's see. I'm just making sure we cross out the extra stuff. Okay. I would not get too specific about ponzo quite yet, because I think that might be the topic sentence for the next paragraph. So I think you kind of just leave off the previous paragraph telling the reader what you're going to do, but don't tell them why yet. Use the why for the topic sentence for the next paragraph. Okay. Okay, so don't explain. Remember, results is not the part where you explain how things work. You don't explain, you don't go into too much detail about the material method, so I would avoid talking about how it works. Save that for the discussion. Okay. Yeah, and what that accomplishes save that for the discussion too. Okay, sweet. So I think this covers our first main topic. Not only did we talk about the results and how to kind of set that up, but the discussion is going to be the same way. So again, you're gonna not only recap the entire overall objective, and we don't need to go into that now because I think that you're kind of pros at this because you did it pretty well with the TLC report. And then you go into, to do this, we start with a Bradford assay to determine the lysate concentrations of our plant samples, right? Okay, to begin or to start or first or meh, great. So that sets up this whole first paragraph, okay? And I don't think you need to go into huge detail again about what these samples are, right? You don't need to explain them one by one. Instead, when you're talking about them, just talk about them. The first time you talk about a certain sample, just call it, and so the plant's grown in a 12-hour photo period, just pretend that's the name of the plant, that whole phrase, okay? So I would avoid re-describing the conditions. And instead, just start talking about them like that. Refer to them as. Okay. And then you state your prediction. Okay. So you kind of like say, all right, so the Bradford assay was conducted. You don't have to restate that it was somewhat BSA and a DF of 2. Just say a standard curve produced a positive trend with an equation of this. So you recap the results. This this data did support or was in line with our hypothesis or prediction. And so you recap the results. You say whether or not the data supported the prediction or not. Obviously, you need to talk about your prediction, and that your prediction is based on this, and there should be some reference. Boom, done, 1 sentence. Okay. And then if you're going to talk about this weird thing that's happening here, you can start with, we noticed after determining the concentration or calculating the concentration, we noticed that the concentration for the negative control was much higher than that of the positive control. And so this was surprising because, and you say your prediction and you say why you have that prediction in mind, so there should be some reference. And the data here then must suggest then that cells in different locations of the plant must produce different amounts of protein. Specifically, cells from the this area must make more protein than cells from the this area. Okay, so specifically, and so you'll figure that out, right? Depending on where I got the tissue for the negative control, where I got the tissue from the positive control, and which one has more concentration. Okay. So a specific data point and a specific detail. Okay. And so this first paragraph, let's do the numbers just for this one paragraph. So really you can see that these papers don't have to be as long as you're writing them. You just have to cut it down and work on being concise. Okay, so 1, 2, as you're describing or recapping the results, this could be probably sentences 3 and 4. And then I would say this right here is probably another 2 to 3 sentences, if that. So let's call it 2 sentences. So it's sentence 5 and 6. And then you need some kind of transition. And the transition sentence here can really be a mirror image of what was in the results. Just change the wording around a little bit and you'll be good to go. Okay. So on average, that's probably pretty good, right? Paragraphs are anywhere between 4 sentences, and really your sentences or your paragraph should really not exceed 8 unless they're extremely concise, short sentences. Anything more than 8 is, you probably need to break that into two paragraphs. Okay, questions on results for this topic on discussion. So this is the time when you have just turned this in. So it should be fresh in your mind, even if it's like, oh, that's not the part I wrote. That's a bad way to think about it. So you should be in charge of all of it. So even if you didn't write the first draft of it, you should have at least proofread that part so that you know what's going on. Okay, any questions on either of these two sections? Results, discussion, Bradford, protein concentration. Okay, let's go. Okay, okay, Madison. Should we mention Kumasi blue? By saying that you're doing a Bradford, you are mentioning Kumasi blue. In the discussion, yes. So, okay, so I would say you'd want to add, you know that you're going to begin, a Bradford is done. This should be one sentence about how Kumasi blue does this, which allows this, which allows this. Very concise, very simple. This is not supposed to be the star of the show. Okay. Great. These are the questions I want to answer. So if you're like, well, I put this, but it's not on the sheet, what do I do? So because we're not going to cover every single detail, right? That is something that's in the expectation. So if we don't bring it up in class, I still expect you to put it because it's in the expectations. Any other questions like that? Okay. Any questions about how to make the figure, how to make the table for the protein concentrations? Any questions you're like, is this too much for a table? Or do we need a table for this? Because I noticed it was in the manual. Am I supposed to add that? Anything? Okay, great. So let's move on to topic 2. Topic 2 is all about the actual Ponzo stain. Right? And we had a really nice transition from protein in a tube to red stain on a little white piece of membrane. All right. So let's see, person number 4. Don't press enter, but please put your topic sentence here. And I will tell you when to press enter. Okay, go ahead and press enter. Let's see here. Okay, so we performed a Ponzo stain. Okay, so the Ponzo stain and then some information that the Ponzo provides. Okay, after initial analysis, the protein lysates were transferred to this. So that I think we covered in the previous sentence, or the previous sentence. So I would get rid of the lysates were transferred, especially if your transition sentence already has that in there. Okay, I would maybe start it with Ponzo staining was performed in order to. You can use way less words. To do this, a Ponzo stain was performed. Okay, so again, here's what we have so far. A couple of pieces of information. So the info that Ponzo provides. Okay, so in order to get that info, Ponzo was performed. Okay. 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 I don't know that the molecular weight has anything to do with what should be in the topic sentence for this. So I would save that and we can bring it in later. But remember the topic sentence, you want it to kind of not be too broad, but not be too specific. Having a molecular weight as part of the topic sentence makes it seem like the molecular weight marker is something of extreme importance in the Ponce stain, which I don't know that it really is so important, right? So some of the things that people are saying that a Ponce, some of the information that a Ponce stain provides is that it confirms the transfer. Okay. Anything else? As we've talked about this for sure. So it not only confirms the transfer, but what is at least one other piece of information that by analyzing the Ponce stain that you can derive. So the Ponce stain, yeah, if you see red, then obviously the transfer worked. But what else? Yeah, ensures that proteins were separated, right? It confirms. And so if you're thinking about it in chronological order, you might even say that it confirms proper, successful, efficient separation of proteins in each sample and successful transfer to the membrane. Okay, so I think that that would be a nice topic sentence. To confirm successful separation of proteins and proper transfer to the nitrocellulose membrane, a Ponce stain was performed or done. You're not, so separation and denaturing are two separate things, completely independent things. Denaturing is talking about the proteins themselves going from quaternary structure to primary, and separation talks about them actually being separated by their molecular weight. So I prefer you to use the correct term. Okay, yes. So I'll let you decide what the correct term is. That's why I didn't answer your question directly. Okay, so that is kind of the topic sentence, and what we've been saying this whole time is the topic sentences for each section are gonna really kind of be similar, not exact. They're gonna kind of be similar in the results in the discussion paragraph. But one thing that you can maybe add into the discussion paragraph, maybe not in the topic sentence, but maybe in the next sentence, is one sentence that describes how Ponce works. The same way that you did it up here, one sentence with how Kumasi allows this to happen, how Kumasi works, one sentence about how Ponce works down here. Okay? So you can see that result really is telling the reader what you did and then telling the reader what you got. The discussion is like, here's why we did it just a little bit. Here's a little bit of background info about this technique. And then let me recap the results and then let me tell you about what those results mean. Okay, so that's a really great topic sentence. And then why don't we go ahead and actually start writing some of the phrases. So what did we say was going to be our template? Very common for science writing is what's common for all the samples. So I think this is an everyone question. So everyone hopefully looked at the Ponce stain, so everyone should have seen something that was common for all of them. Everyone, you don't have to wait to press enter. Just start listing things that you saw that were common for all of the samples. I'm going to pull up the data right now too, so that's what I'm doing. Just give me a second. Okay, let's see. This is Wednesday. Okay, so let's see what you guys wrote. So all of them had in common, let's see, we start at the top. All 3 lanes displayed bands at 13 to 15 and 55 kilodaltons. Okay, I would say that, sure. But you want to start with what is the most obvious thing that you see. And Francisco, I would say what you wrote is the most obvious thing that can be applied to all of the lanes, right? So every single sample, no matter what kind of sample it was, no matter what the growing conditions, there was pink or reddish stained bands from 10 kilodaltons to 75. Okay, maybe that's the top part. Okay, let's see. Yulica's saying that there's a band at approximately 13 and 55. Yeah, so this is getting pretty specific, but let me ask you, for the ones that said there was bands at 13 and 55 kilodaltons, let's write it down, because I'll let you, this would be writing tone. This is writing style. So let me go back. I have so many things plugged in. Okay, so in common, you start broad and then you get more specific. So the most broad observation that you can make is that all 3 displayed bands from some certain range from some certain KD to some certain KD. Right, so I'll let you figure that out. That is the most broad observation that you can make. And then people are also saying that in the 3 lanes, they did see bands. You're pointing this out, so I'm gonna put this in parentheses because it really does depend on how you write. You also saw a band approximately some KD and some other KD. Now just be careful with that, because if later you're going to say that there was barely any protein at these markers, then I'm going to say, then why did you make such a point that you saw it in the ponzo if later you're like, oh, it was barely there? So don't contradict yourself. Don't set yourself up for contradiction. Okay. And Kristen, I think that what you're saying too is true too. So there was many bands, see the trains coming, many bands from this point to this point. Okay. And so what does that mean? That there were proteins of all different sizes and amounts or concentrations, right? So that, I think, that's a conclusion that you made from seeing a bunch of red stain. Seeing a bunch of red bands is the result. What does that mean? That goes in the discussion section. Okay. Right. Okay. Great. Okay. Alrighty. So that's a thing in common, and we've matched it with whatever discussion goes with it. So I guess from here, the one thing that you need to remember to do is, what was your prediction for this? Did you expect, so this is a for everyone question. It's just either yes or no. Did you expect to see bands of all different sizes and shapes and locations on the gel? Yes or no for all 3 samples? Cool. So your prediction, and then you base it off of some kind of published work. And then you can say, given that ponzo stains all the proteins non-specifically, blah, 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 we expected to see this and that. And in fact, indeed, the data did support this, or yeah, something like that. So data support, the answer is yes. No further explanation needed. There's nothing weird, so don't worry about it. Okay, now, and then you finish off with, this indicates or further supports the notion that plant cells make, at any given point in time, plant cells make proteins of all different sizes and variable amounts. Alrighty. And so now you can get into the discrepancies or the differences between the 3 samples. So, question for everybody. What is the first sample you presented in writing? Next question, everyone, why? Why did you talk about positive control first? Cool, good. So you don't need to tell the reader that in the report. We're talking about the positive control first. As you can see, it's the first in the lane. No. It's just, no. It's just unwritten law. This is what you do. Cool, okay. So you looked at the membrane, you're like, okay, besides the molecular weight marker, we're not gonna go into detail about that because that's not data. The first thing that we are gonna talk about in terms of result is whatever's in lane 1, and whatever's in lane 1 is, oh, it's positive control. Okay. So the positive control, now let's go with, so you're telling me it's in lane 1, so I'm gonna write that down in parentheses for now because I'm gonna say something about that in a second. Okay, so it's in lane 1. Great. This is an everyone question because I think everyone can answer pretty quickly. So what are some of the phrases that you use to point out the results of the positive control? Okay, everyone should be typing something in the chat. Okay, so I see a couple different ways to describe data. You're describing it by thinner or thicker bands, okay, and you're also describing it by size. And size, we're talking about kilodaltons. So I think to be the most specific, you're going to just basically say that the positive control had a very thick band at wherever you're telling me. That maybe you can start with how many band. Oh no, we're in the Ponce stain. Okay, so we already said that there's a bunch of bands, but the thickest bands at approximately, do not write at 55 kilodaltons because you don't know that it's 55 kilodaltons. You're just assuming that it is. So you can say approximately 55, or you can say slightly larger than 50 kilodaltons, but no matter what, you're saying as compared to the molecular weight marker. Just, you don't have to do it every single time you talk about the results, but just sprinkle it in that you're actually coming up with these numbers based on some known standard. So the thickest bands at whatever size you observe them to be at, and then maybe just once, right? As compared or versus compared to the molecular weight marker. And again, as a reminder, we're taking notes in this class. You are going to be writing the word at. You're going to be writing the word approximately. You're going to be writing the words plants grown in a 12-hour photo period. Okay. So I think like a good rule of thumb is for every major set of data, you want to at least refer to the plants by what they really are, at least once, and then the rest of that set of data, go ahead and use the phrase positive control. So I'm gonna put a line through this, and I'm going to hopefully remind you to use a more specific descriptor of the plants. Okay. If you want, again, to put the word positive control in parentheses, that's absolutely fine. And then for the rest of that paragraph, if you want to talk about them as the positive control, that is fine. Okay, and that might make sense with your writing style. Okay, great. So, and usually when, yeah, okay, never mind, scratch that. All right, so the next sample, I'm going to abbreviate it by just calling it negative con, but now you know what I'm talking about. Let me actually move this down. Okay, so this is an everyone question, right? What is the data that you reported for the negative control? And I'm going to remind you what you should be doing here. Stating the data and building it into your writing sentence, how you're comparing it to control number 1 or sample number 1, whatever that may be. Okay. Everyone should be typing in the chat right now. Okay. Okay, we've got some variation here. Okay, so it starts here. So I see thick bands at approximately 37, 10 to 15. The lane that had a sample with no photo period had a thinner. Okay, so great. Thinner bands. We're not talking about 60 minutes of light right now. We're only talking about this sample and the one that we've already talked about. We have the lowest. Okay, cool. So I think that you can't say intermediate. Intermediate implies there's 3 things. You haven't talked about 3 things yet. So right now you're only talking about the sample that we are talking about and the one that we have talked about. You cannot compare it to something you haven't yet talked about. Okay, so at this point in time in the report, there's only 2 samples. So you can't say anything about intermediate or first, second, third, or you can't compare it to the 60-minute light because you haven't even brought that up yet. You always have to establish your controls first, show the reader that the controls worked, that they have opposite results so that you have established a positive control, you have established a negative control, everything's gravy. Now you can talk about the experimental. So using that rationale, the negative control, what you want to do is, since in the previous sentence you talked about the positive control having thick bands at these so-and-so places, that is the first detail you should be talking about for the negative control. So you want to compare to those thick bands in the positive control. And the way that you compare it, you're telling me, if I look back at the, I know what it kind of looks like. You're telling me that you use phrases like thinner, but I think it's valuable to also remind the reader thinner at, so something like this for plants. So positive control, so plants grown in a 12-hour photoperiod display bands at blah, 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 very thick bands at these places. In contrast, right, because it's in contrast, or unlike the positive control, or whatever it is, in contrast, plants grown in absolute darkness or the absence of light had much thinner bands at 55 and approximately these 2 band sizes. Additionally, if there's other stuff you want to say about the negative control, go ahead and say it. So you want to start a sentence with something like unlike, or in contrast, or conversely, any kind of phrase that provides context, right? You want the reader to really understand that the data for the positive control is opposite that of the negative control. So you want some phrase that provides context, the negative control, thinner, and then you have to include those sizes. And then additionally, if there's any other pieces of results that you find absolutely necessary to point out because you're going to discuss them later, or you think it impedes on your ability to make conclusions about the Rubisco in the final stages, anything like that needs to be pointed out here. Additionally, we also observed if that exists. Okay. All right, so okay, so here's a question. Should we mention the approximate number of bands? I would say if you could actually count the bands, the answer would be yes. But I'm looking at the picture right now, and to me, I can't really see it. Like, I definitely see this as a band. I definitely see this as a band. And yeah, there's probably a band here, probably a band here. So here's how you can think about whether or not you want to point that out. Do those bands have an impact on your final conclusion of this paper? If the answer is no, just kind of like say many bands of different sizes. I think what's most obvious here is that there's a very thick band here, a very thick band here, and then just a bunch of banding. It also seems like there's, yeah, you can maybe make out a couple distinct bands, but really it looks kind of like a smear, but that just indicates that there's so many different proteins of so many different sizes that all the bands are blending into one another. Okay. Alrighty. Yeah, Stephen, you can in a second. So I'm going to do that at the end. Okay. We'll do the results, the discussion, and then questions about those things, and then questions about the figures. Okay. So before we get into, let me go back to my notes. Before we get into the last sample, I want to take a time to remind people that they should be referencing their figures while they're talking about results. So all the things that the samples have in common, let's see if this is figure, what would it be? Probably figure 2. Let's pretend it was figure 2. Figure 2, we know that figure 2 is going to have 2 panels. You were asked to make a model of the ponceau, and models are always panel A, and because it's kind of like your prediction. And then you're going to have your actual results as panel B. So if you are talking about actual results from all 3 samples, you need the reader to look at the entire panel, so panel B. So here's where you would just write at the end of this sentence, figure 2B, or whatever number the figure is. Okay, and then you can even help the reader by helping them really direct their eyes at one specific part of the panel. So if you are talking only about one lane, data in one lane, that's why I put it in parentheses. If you have made your figure and you have labeled the lanes 1, 2, 3, when you say the results for this, at the end of this sentence, you can actually write lane 1, comma, figure 2B. So not only are you telling the reader, go to the figure with, go to the page with figure 2, look at panel B, look at lane 1. There you have it. But this sentence down here, you're comparing 2 lanes. So you can say lanes 1 and 2, figure 2B. And honestly, I've seen figure 2B, comma, lane 1 and 2. I've seen it both ways. Both ways are fine. Just pick one way and stick to it throughout your entire paper. Okay, if you're comparing more than 2 lanes, don't use the word lane 1, 2, 4, 5, 6. Just call it figure 2B. So this or this applies for comparing 2 lanes. That's it. If you're comparing 3, don't even bother. Okay, so don't forget to do that, not only in the results, but in the discussion. That's where I'm noticing people are forgetting to add references, where people are forgetting to reference their own figures and tables. They're just like writing a bunch of stuff in a discussion. I'm like, okay, well, where's this coming from? Or where's this data? Which figure am I supposed to look at? Okay, every time you mention results, even if you're recapping them, you got to send the reader's eyeballs to go look at it again. All right. Okay, so same thing applies for all of these sentences in the same order. So you will, and you don't have to do this sentence by sentence. If you can write it so that you can make your prediction about both controls and why and references, you can go ahead and do that. And then you can recap with figure 2b, or maybe it's even figure 2b, right? Why would I say it could be both? Because the predictions, you should be referencing figure 2a, right? That's the whole reason I had you do this model, so that you can see what it would look like if it was perfect. And if you're going to be writing about it, might as well tell the reader, like, yeah, this is what it would look like if it worked perfectly. So just use the appropriate figure references as you're doing this. Now, let me ask, so I'm going to revise this little arrow, because you could potentially, depending on writing style, you could potentially squish it into one nice, concise sentence. Now, if it doesn't fit like that for you and you're a really wordy person, then just do it in 2 sentences. Okay. But what is one thing? Okay, so yeah, we're talking about the Ponce stain. What does it mean? So this can mean a couple of different things. That, what did we do this for in the first place? To confirm separation of protein, successful transfer of the membrane. So you want to actually write that. Hey, this data in fact suggests that the separation was successful, right? As evidenced by a huge display or a variety of bands, that the separation and the transfer were successful. And just a recap of the evidence, right? Indicated by various red banding in all samples of all different sizes for, or red banding of all different sizes for each of the samples. Okay. So this is the meaning. And then you provide the evidence. So in this case, it would be bands, all sizes, each sample. Okay, what other hints? This is for everyone. Don't press enter, but I want everyone to make a guess. Here's the prompt. We are doing this entire study to look at the effects on Rubisco. Specifically, we are analyzing 60 minutes of light. Okay. Now, can the Ponce provide any kind of clues that would help us get to the major conclusion that we're going to make? And if the answer is yes, just type, don't press enter, just type in what other clues or possible information can the Ponce provide. Don't press enter, do type in your guess. All right, enter. Ooh, good job. Let's see. Okay, so a couple of these statements are true. Ponce can do this, but they don't really relate to the objective of the study. The objective of the study has to do with Rubisco. Okay, so basically the question is, can this Ponce stain tell us anything about Rubisco levels? And people are saying yes. amount of Rubisco expressed, okay? If Rubisco is known, seeing the bands at those KD in the Ponzo can hint that there is in fact Rubisco. Yes, okay? So you have to kind of piece things together. We know the subunit sizes of Rubisco. So if this whole project is about Rubisco, wouldn't you want to point your eyes, and you kind of already did it because you're like, hey, the Ponzo stain has all these thick bands at 55 and approximately 13. Why were you telling me that? Who cares? And you know that the reason why you're telling me that is because those KDs are important. Why are they important? Because those are the sizes of the subunits. And so it's when you get to the discussion, you can mention this to the reader at this point, right? So these results also may suggest or may also indicate levels may, okay, this is the key word, may indicate levels of Rubisco between the three samples. And then you want to recap what that conclusion is. You're going to look back. You're going to say which of these samples has the thickest bands at whatever special size I should be looking at. And that means that this Ponzo stain may suggest that sample 1 may in fact, no, not, we don't want to say in fact yet, may produce higher levels of Rubisco than la-di-da-di-da. Okay? But you always want to focus it on the experimental group, because that's the question mark. The positive and negative controls are not our focus. They are there as tools, okay? So may suggest the levels of Rubisco in plants exposed to 60 minutes of light are higher or lower than blah, higher or lower than, well, I guess equals is another option, right? Levels of Rubisco in plants exposed to 60 minutes of light are approximately equal, higher or lower than whatever sample. Paloma, please. And then you compare it to the other control as well. It's important to say may because the Ponzo is non-specific, so you can't make a definitive conclusion. Okay. Hannah, you bring up a good point too. What can it also show us? Not really has anything to do with Rubisco, but yeah. If we're looking at the variable 60 minutes of light and there's some weird stuff going on, it also may suggest that 60 minutes of light exposure has some kind of an effect, positive effect, negative effect, or no effect on protein production in general. Or if you see a bunch of bands that are like higher up on the gel, you say like, oh wow, 60 minutes of light actually makes a plant produce large proteins. Or if you just see small bands, wow, 60 minutes of light impacts the plant's ability to make large proteins, negatively impacts, inhibits, whatever. So this would be an example of critical analysis. This is what opens the doors for more research. Okay, and it's, shit, sorry. It's all based on, okay, she got chips. It's all based on, focused on 60 minutes of light. Okay, we're not going to be saying anything about the positive and negative control. All right, when you're talking about this stuff, you're still going to be referencing the figures, the appropriate figures as necessary. Okay, so if you're talking about all three lanes, you might here just write figure 2B. If you're talking about 60 minutes of light, again, if you're talking about all of them or just one lane, just put the correct figure and table references when necessary. Okay, last but not least, your experimental sample. You're not going to use experimental sample. You're going to say plants grown in or exposed to 60 minutes of light, displayed a band. And here's where you just compare it to both of the controls. You can do it in one sentence, or you can do it in 2 short sentences. You can use things like greater or thicker band than the positive control, but thinner than the negative, or you can say much thinner than the positive and approximately equal to the negative. However you want to say it, all we're going to take time to say here is that you need to compare them very clearly to both controls. This can be 1 sentence, 2 sentences. Use linker words, context phrases. Provide context. Make it super clear. And that's it. And so this one would probably just be figure 2B, because you really are comparing it to all of them. You're comparing multiple things. Okay, so now, Stephen, I'll take your question about the figures for this.
So I put the KDA values on the left side next to the molecular marker, and I was wondering, when I do the result labeling, should I refer to that as the y-axis?
It's not a graph, so you don't need an x or a y-axis here.
That's what I thought.
Yeah, so if you have, let's say, here's your piece of paper. You have 2 figures, 2 panels, I should say. So here's my figure 2. You have some title that encompasses both panels. You do not have 2 titles. Okay? 1 title that describes both panels. Okay, so blah, 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 whatever that title is. And then in sentence form, I'm going to write it in bullet points, but you should be writing it in paragraph, like a regular paragraph. The first thing that you want to start with in this figure caption are the details that apply to both panels. So details for both panels. This includes a brief recap of the methods, something along the lines of what you said up here, okay? Protein lysates were run on an SDS page and transferred to a nitrocellulose membrane for Ponzo staining. That's it. Nothing else. It needs to be 1 short sentence. And so now as the reader is reading this, they're saying, okay, so I'm staring at a nitrocellulose membrane that has been stained with Ponzo. Got it. Okay, so that means lysate on a gel, on a membrane for Ponzo. Okay, so, okay, cool. Now I know what I'm looking at. I'm looking at nitrocellulose membrane. There's those Ponzo stain bands on it. Great. All right, then what? And then you say that, I don't know, something like the banding pattern or the Ponzo stain was compared between plant samples grown in. So somehow you have to work in your plant growing conditions. And I know you're like, I already did this in the methods, I did it in the result. Yeah, you're going to do it again in the captions. Okay. The growth conditions have to be built into the caption. So if you are doing any figure with multiple panels, this is one thing I did not see, and I was surprised that I didn't see it. Panels need letters, just like an actual real paper. A, B, C, lowercase I, like Roman numerals, whatever it is, they need letters. And so you have your panel A, and then you have your molecular weight. I'm doing it all in black. Your molecular weight marker. Stephen says that he also put the KDs over here. So Maybe he has KD up at the top and this is like 100, this is 50, 25, whatever. Fine. Those numbers should reflect the actual molecular weight marker that you did use. Okay, so that should look almost identical. And then you have, sometimes people like to just put a little line to indicate where the lanes are. And then they say 1, 2, 3. Okay, you have to label the lanes 1, 2, 3. If you're going to be writing about them like this, lane 1, figure 2b, then yeah, those lanes have to have actual numbers. So other people are like, what if I put positive, negative 60? You can do that, but then you can't use this format. You can't put lane positive, lane negative. It doesn't work like that. So choose however you want to do it. Okay. So, and then you have your, whatever your predicted model is. Great. But here's the point. The point here is that you don't write things like the plants grown in the positive control condition are in lane 1. That's not the format of captions. Instead, you would say, you would just start with the phrase plants okay. Something like you analyze plants or Ponzo staining of plants. Plant lysates were separated on an SDS page, then transferred to a nitrocellulose for Ponzo stain analysis. Lysates include plants grown in a 12-hour photo period. Okay, so there's a lot of different ways to write this. I should not see 20 reports that start the same way in the caption. So I'm going to write this, but you can't use this one. Lysates include plants, P sativum specifically, plants grown in whatever they're grown in. And then here in parentheses, this is where you write the phrase lane 1, comma, in the next condition, lane 2, or in the third condition, lane 3. That's how you write it. Sure, use it thesaurus, do whatever you want, but this cannot be the same. Okay, and so those are all the details that apply to both panels. Yes, right? So no matter if you're looking at this lane 1 or this lane 1, it's still plants grown in blah, blah. If you're looking here, it's still plants grown in la-di-da-di-da, right? So now you can differentiate between the two panels. So let's make this a little smaller so we can fit more stuff in the caption. Okay. So once you're done detailing the information that applies to both panels, now you can write the difference between the two. So whatever A is and whatever B is. So what is A? What would you describe A as? Somebody just tell me in the chat. What's A? Panel A. This is panel. Panel A, panel B. Okay. You can write something like, what you're basically doing, you're comparing the two, right? Comparison, and you can make this an actual sentence of predictions. And then here's where you write panel A. Maybe you want to bold face this. A comparison was performed, a comparison between the prediction, panel A, and actual data was performed. Something along those lines. And that's the only difference between those two things. One's fake and one's real. Okay. Okay, cool. So now, are there any other questions about making a figure when there's multiple panels? Any questions about the caption? And a couple of reminders, because I saw many people not do this. The caption needs to be justified text, means aligned on the right, aligned on the left, and single spaced. Don't forget. Okay, cool. So if there's no more questions, that's the pond system. Does the title go on top? I'll let you look at the expectations for general lab reports, okay? That's all highlighted there. There is a difference between figures and tables. Okay, so pond sustain. Any last questions about any part of the pond sustain? Results discussion figure. All right, so I think let's go ahead and take a break. So let's do 8 minutes, 7 minutes. Let's be back at 12:30. We'll do the last topic. And I'm giving you a break because I think you deserve it. You've been working really hard and I really appreciate that. But I don't want you to lose focus. So take these 8 minutes to get we'll give you 8 minutes, okay? To get a drink of water, whatever you need to do. So when you come back, you still have that same energy and we finish out strong, because the last segment is the important part. This is the paper. This is what is going to be the take-home message. So let's say math, 12:31. See you then. In the last section for both the results and the discussion, we're going to be pointing out, then explaining what the immunoblot is and why it was done. So I think that let's go ahead and put person number 4 on the spot this time. So person number 4 from each group, please don't press enter until I tell you. Go ahead and type the topic sentence for the immunoblot. Remember to try to be specific. Don't hit enter. Okay, go ahead and press enter. Great, I like the specificity here. Okay, so definitely to compare the amount of Rubisco in the 3 protein samples, close. So we're not comparing Rubisco samples. We're not comparing 3 protein samples. We are specifically looking at, see if anyone has it. What specific condition are we looking at? Remember, the other 2 samples are not real samples, they're controls, so we don't count them as samples of analysis. Okay. To determine whether or not, or to determine whether 60 minutes of light has an impact on Rubisco expression. Okay? Something about 60 minutes of light. That is the only condition we're actually analyzing for an answer that we don't know. Okay. Rubisco levels. I like that because this is kind of like the big bulk of it. We haven't talked about P sativum in the results section for a very long time. The first time we brought it up was right at the beginning. So when we end this results section, that's a good time to just one more time squeeze the species name in. Okay. All right. To look at the levels, to compare the levels, to analyze the effects, so an immunoblot was performed. Great. So The topic sentence for the discussion is probably going to be pretty similar, but there's one thing that we can make it so that it's slightly different and a little bit more explaining. So is there anything else that we can add to maybe some of these things here that can be a little more detailed or a little more deeper? So we can't really talk anymore about 60 minutes. It is what it is. What about Rubisco levels or Rubisco? Can we get a little bit more specific about Rubisco? This is for anyone that can think about it. Right, the specific subunit. So we can specify immunoblot by saying, by blotting with an antibody specific to the correct subunit of Rubisco. Where are we, if we had enough plants to do this, where was the experiment supposed to be taking place? What part of the plant? Okay. And then since the discussion, this is your last paragraph of the discussion. Here's where I think that you want to, one more time, remind, this is the last reminder the reader gets, to analyze how 60 minutes impacts Rubisco expression levels. An immunoblot was performed with an antibody specific to the large subunit. Sorry, I think we should have rephrased it. To analyze the effects of 60 minutes of light on P sativum. And then I don't want to give it away because I know everyone's just going to pause the URL and then just write it exactly how I'm saying it. But you can definitely build in that for this last entire thing that you're going to be analyzing the effects of 60 minutes in the leaves of the plant, the specific plant, using an antibody to the specific subunit of Rubisco. And then you want to remind, so that's your topic sentence. And then I think that you want to remind the reader one more time, the growing conditions. If you are a very concise writer, you can probably fit this into the topic sentence. But don't force yourself if it's really hard to do. Or it just feels like too long, it feels like a run-on. Don't force yourself. Okay. So that's your topic sentence. Do you see how the results was a little bit more broad, but the discussion was a tiny bit more explaining? So that's where we're at there. And then you want to use the same rationale as you did, the same approach as you wrote the previous paragraph. So in the previous paragraph, we started with all the things that we found in common between the three samples. So I think this is an everyone question, but there is a certain question. So Rachel, what is it?
Do we mention at all that we took samples from the stem in this part of the discussion for the L60, or no, because it wouldn't be present in the stem?
I think that if you need to use that detail to describe unexpected weird data, that's when you would bring it up.
Okay, thank you.
Okay, so let's start with an everyone question. What is one thing that you found similar for all of the samples? Okay, so it looks like For all the samples, if we're talking about results, we're just talking about purple bands. For all the samples, no matter what sample it was, it seems like people are seeing a purple band at approximately 55 kV. Okay, great. For all the samples, perfect. And keeping consistent with the format that we've been using, we know this will likely be in some figure, whatever figure that is, but it will be in panel B, right? So the same way you have your console results, where you have your predicted and your actual results here, you're gonna do the same thing for the immunoblot. So this, and since you're talking about all three lanes, just make sure that you have whatever the correct number is with panel B. Okay, everyone's putting at 55, but I know you mean approximately, or I know you mean slightly larger than. Okay, so I'm going to clarify one thing. They did not all have bands where this Rubisco subunits are supposed to be, because if I look at the actual data, I do not see bands at the 13 kilodalton mark. So that is slightly inaccurate. So literal details, I know we use the word literally like very incorrectly in today's day and age, but literal means word for word. So if you don't see both subunits, you can't say that you see the subunits there. You can't say that. So literal words, details matter for science writing. It's okay. It's no need to apologize. It's fine, but I want to make sure that when you leave today, you're like, well, I said it and she didn't say it was wrong, so I guess it must be okay. I'm just making sure that we point out the things, the inconsistencies and the inaccuracies. Okay, so let's go back to this thing. All right. Same idea though. So if you see this in all of the samples, you want to talk about it in the discussion. Same template, the prediction. So you want to, in whatever order makes most sense, but you better have a recap of what you saw, predictions with a reference, whether the data support it or no, and then what that means. So if it's yes, you just say this suggests or this indicates, and letty-doo-doo-doo, okay? So this is the easy one. If no, if you did not expect to see that, you need to explain, okay, well then what does that mean? And so I think most groups predicted to have low amounts of Rubisco in plants grown in the darkness. But I don't know, different teams may have different hypotheses for what they expected to see in the experimental sample, for the experimental sample. So this is gonna, here what happens. This is gonna depend on what your hypothesis is. And I'm just gonna give you a little hint or a reminder that Rubisco functions in two cellular processes. So you can use this to your advantage. If you have weird results, this might indicate something about that second process that we haven't really talked a lot about. Also, this is a big thing, is you spent so much time talking about the Ponzo stain, and then you went and told me that this potentially could provide some information about Rubisco. This is your opportunity to say like, oh my God, remember the Rubisco of the Ponzo staining? Actually, the results look the same. Like this big thick band that we're seeing at 55 looks really similar to that 55 KD band that we saw in the Ponzo stain. So you need to have you need to build it in somehow, whether or not this data is in line or complements or is similar to the Ponzo stain. Yes or no? Yes, cool. Then use a cool word like, oh, as expected. That's not a word, it's two words. Okay, because you kind of did expect that, right? If you saw something in the Ponzo that was nice and thick right around the supposed subunits of Rubisco, and then you in the immunoblot, you actually see that fat band, you're like, okay, cool. So what does it mean? it means that the thick bands or the bands at approximately 55 kD in the Ponce stain were in fact the representative of Rubisco subunits. So it was Rubisco. If the data does not agree, then you need to tell me what you thought, what those bands were in the Ponce. So, okay, then what does it mean? All right, don't forget that part, important. Because it's like saying one conclusion using two different techniques to kind of back your statement up. So question, should we make a brief statement comparing the immunoblot to the Ponce stain and the results as well? Absolutely. You can even start with, similar to the Ponce staining pattern, we observed a band at approximately 55 kD for all samples in the immunoblot. Bring in or compare to Ponce. Okay. All right, so that's the thing in common. Then we have, and so if you are comparing it to the Ponce stain, then really this should be figures 2B and 3B, right? You need to tell the reader to look at two separate things. So let's go with the positive control. Okay. And at this point, because it's the last sentence in the results, I would maybe use the actual phrase of what the sample is. So specify the sample. Okay. Because up here we did it at the very, very beginning. Where to go? We did it kind of at the beginning or throughout the paragraph. And then once we were comparing to, we started using positive control, positive control, positive control. That's fine. This is the last impression. So you want to specify the sample, and then through the rest of the paragraph, you can go ahead and use positive control again. Okay, so let's go with, we're back to person number one. Person number one in each group, tell me the actual data that you saw just for the lane one in the immunoblot. And you can describe them in thick versus thin, but just go ahead and you don't have to wait to push enter, just keep putting things in that you saw and the phrases that you use to describe them in your actual report. Okay. I'm gonna look at this real quick. I'm gonna find it. Okay, let's see here. Oh, wow, okay. All right, so I'm looking at the data too. So I think if you think about how we started the other sets of data, we started by talking about the 55 kilodalton marker, or the band at approximately this location. So I think it makes most sense because we like to be consistent and it's the same thing every single time in the same order. I think it's important to actually start with that. And there was a lot of bands, but this one happened to be the thickest. So again, you're describing it by thin or thick, and you're describing it by size. Maybe if you haven't done it in a while, then you can say compared to the molecular weight marker, as compared to, or as interpreted from, whatever. Okay. And then you always want to start kind of presenting the most obvious data, which I think was this. And then you want to move on to the other things that can also be kind of weird, right? So it's the most obvious, the less obvious, and the really strange, weird, oh my gosh, what happened? So there's not really less obvious, it's like super obvious and weird stuff. So the weird stuff, people are saying that they saw 9 bands, people are saying that they just saw multiple bands, but what is important is however many additional bands, and if it is, did it surprise you? If so, surprisingly, we also observed X number of bands, so use your context phrase that alludes to the fact that this was not expected, that it was a surprise, that you're like, oh crap, okay? You can provide a range, or if you really think that providing the exact numbers, like if you're going to use that in your discussion, exact numbers, then you can say, let's pretend there was 4. 4 additional bands were observed at 20, 30, 40, and 45 KD approximately. That's all you need to say at that point. If you're just needing to say that they were all between 20 and 45, that is okay too. So that's going to depend on how you write your discussion. So the options are you provide a range, or you specify, this is kind of counterintuitive, specify approximate KDs. Okay. The context is what is happening here. Okay. All right. So, and then you might want to just mention, at least for the 55 KD, was this similar to Ponce, yes or no? Right? You might want to just say this thick band at approximately 55 was very similar to that observed in the Ponce statement. And then you have, again, figure lane 1, figure 2B, and lane 1, figure 3B. Okay? Okay. Great. And that's it. So when you're doing this in the results section, or the discussion section, so let's move this into the space. Okay. You use the same approach. Did you expect a very thick band? You do each of these one at a time. Did you expect a band here? If so, why? Recap the data. Did the data support, yes or no? If the answer is yes, great. This indicates that plants grown in a this and that condition do express a high level of rubisco large subunit. Okay, and so it's pretty like intuitive if it's going to be if it's going to express the large subunit, it's probably going to express the small subunit. So you can just say, wow, rubisco, woohoo. Okay, if no, you explain that. But here's going to be the hard one. Not hard, because we've talked about it. We've talked about reasons what bands smaller than the predicted size could mean. We've talked about what bands larger than the predicted size could mean. So, but you still use the same thing. Surprisingly, some kind of context. We did not expect bands at wherever that is. And by you saying that, you are recapping the data. Now you just have to have a brief reason why you did not expect that. And I think that just kind of goes hand in hand with, well, rubisco is made up of 2 subunits, any other size, what the hell's going on? Okay, so your prediction, and it's possible that these predictions could be built into one sentence, right? You don't have to restate it again for that next little smidge of data. You can just say it once, so make sure you're not redundant. And then obviously, if you're providing context that this is not expected, you need to explain what this means. That's the speculate. So what does it mean? And we've talked about this. So this is when I'm going to remind you to go back to previous workshops and look at the videos as to that would describe why you would get smaller than your expected KD. And then maybe how you would fix that. Okay, and then it can be here, it could be here. Somewhere you need to talk about concept. So we're just going to put red stars to remind you that somehow you have to build it in so that it feels like it flows nicely. Okay, but you're comparing it to concept. Okay, so looking back at the immunoblot, we know that we're going to do the negative control next. This is an everyone question. What is the data that you saw for the negative control? Please describe it. Okay, so there's one very obvious band at approximately 55 kD. It was thinner than, so that's you're comparing it to the positive control. And in doing that, you have set up your ranges, right? The high and the low, positive control versus negative control. Okay. I think someone I saw put smaller band. Be careful with that word. Smaller indicates a lower KB. Smaller does not mean thinner. Okay, so for describing the thickness of bands, you can use thickness, thinner, or more intense, less intense, but you can't use smaller or larger. Okay, and so I don't think we need to get into this here because I think people are hopefully starting to see the pattern of how to do this properly. And then don't forget, somehow building this in, were these results reflected in the Ponce stain? Yes. And if the answer is yes, we're almost done, guys. If the answer is yes, then you can actually make a conclusive statement that the bands originally thought to be Rubisco subunits in the Ponce stain were in fact representative of those subunits. Okay, and then last but not least, this is your huge take-home message. This is when you absolutely definitely want to make sure that you refer to the sample by the actual condition it was grown in. And you want to, if you didn't do it already, it looks like you did it, but this is kind of your last sentence of the results, your last sentence of whatever this thing is, the discussion. If it was me, I would be as official and complete as I can. So I would actually write, P. sativum that was grown in the dark or only exposed to 60 minutes of light before harvest, protein harvest, and then you describe the data. So we're not going to describe the data here, but we know how to do it now. You're going to compare it to the positive control, then to the negative control, and highlight any other unexpected data. So if that data really was opposite what your hypothesis was, you want to point that out, right? If it was in line with that, you can say as predicted, or somewhat similar to our predictions, we found that P. sativum exposed to 60 minutes of light displayed a thicker or thinner band, or approximately the same thickness of that, whatever it may be. But here's where you really want to use like all your official formal words. And last but not least, definitely compare it to the Ponce results. That's it. Similar to what was seen, or similar to the observations made in the Ponce stain. Okay. And then the way that you want to finish your discussion, obviously, you're going to do the same thing that we've been talking about: predictions, recap, references, la, 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 la. But you want to make your final conclusion, like the entire answer to your objective, the whole entire thing, you want to very clearly say, collectively, this data suggests 60 minutes of light, and then you make a statement about 60 minutes of light. You're not saying Rubisco levels are dependent on light duration. We're not really looking at that. We are specifically wanting to know about 60 minutes of light. Is 60 minutes of light enough to induce expression of Rubisco? So it needs to be, is there any Rubisco? Okay. If so, are the levels more similar to plants that are grown in the darkness or plants that are grown in natural states? So something about the levels. And if Rubisco is being expressed, what cellular process? That means 60 minutes of light has an impact, maybe indirect, but has an impact on the rate of certain cellular processes. It could be 1, it could be 2, depends on what you come up with your team. Just better make sense. Okay, and then, so this is kind of like the boom of the whole report. And then you make one final conclusion, making it a little bit more broad. How can you use this to conduct the next study? Do you want to look at a time course of light? Do you want to vary a different growing condition? Do you want to look at a different species? And what kind of relevance does this have to either economy, society, humanity, medicine? Who cares? Yeah, it's just a lab in class, but still, pretend like it's real. So what is the relevance, the significance to the world, some part of the world? And then if you could do the next experiment, what would it be? Great. Any questions? Oh, the last little pieces of advice I have for you for the figure. The format is the same. Panel, panel, one title, stuff that applies to both panels, and then stuff that applies to each individual panel. But in your figure, you want to have specific product information. So that means don't just say, oh yeah, this is an immunoblot. Remember, it's after the nitrocellulose was destained, it was subjected to immunoblotting with various antibodies, including a primary antibody at 1 to whatever. But you don't use this phrase. You use the actual product name and then the information in parentheses, just like you did in the methods. You don't have to include any of the washes, any of the buffer, any of that. But these things right here, important. Primary antibody, secondary antibody, and the substrate. That needs to be in the caption. Don't lose easy points. Okay, now questions on this section here. Okay. Cool. All right. So that was a really good job by everybody. And I really, really hope that leaving today, you feel very confident about the changes that you can make to your own paper. I hope that you leave feeling confident that now you can provide an effective and an impactful review or an evaluation of someone else's report and help them make it awesome. And I also hope that you feel that this is doable. Each person, if you are in a team of 3 to 4, if you block out, you don't have to do it all at once, but I think that the revisions on your own draft, if you put your heart into completing the rough draft from start to finish, then I would guess that this is probably about 5 more hours of work. This is not due until the 28th of this month. It is currently St. Patrick's Day, it's the 17th. So you have 11 days to do this, but I realize the next week is spring break. So there is a small, just like the TLC report, there's a small extra credit incentive to get it done on the earlier side of spring break. I think for you, I have it if you do it turn it in by Monday. And I still think that's really doable. I think that if it's only Wednesday, I think that if you worked on this, you know, one day for an hour and a half, another day for an hour and a half, and then just leave yourself time to kind of go over the entire paper from start to finish, I really do think that every team can get this done by the 22nd. And in doing so, earn 3 extra credit points and not have to worry about it over spring break. So good, I'm glad that it was helpful. So I encourage you to try to do that. And if you don't want to, that's okay. You have until the 28th, so you can still spread out the work. It's just if it was me, I wouldn't want that looming over my head. Okay, so make sure you have a peer eval partner. And give me a second.